Good afternoon, everyone. Hello. I'm Jane McIntosh. I'm the Chief Philanthropic Officer at the Alzheimer's Drug Discovery Foundation, and on behalf of our Board of Governors, our Board of Overseers, and our CEO, Mark Roithmeyer, I'm delighted to welcome you to that was really introduced today, which is a new scientific symposium series that was really inspired by Heidi and Tom McWilliams. Tom is co-vice chair of our Board of Governors, and Heidi is a co-chair of the 5th annual Hope on the Horizon Palm Beach Dinner, taking place on March 3rd this year. I'd also like to acknowledge Nancy Goods, who's here, and she's co-chairing that event, along with Heidi and Bonnie Lautenberg. This is the first of 3 lectures we will present this season. We have some brochures out front about the program, and also we have a handful of save the dates for the Palm Beach Dinner as well. And now it's my pleasure to introduce my colleague, Dr. Howard Fillett, who is the co-founder of the Alzheimer's Drug Discovery Foundation and our Chief Science Officer. Howard?
Oh, thank you, Jane. And let me thank all of you for coming today, and I particularly want to thank Heidi and Tom for making this possible today. Thank you very much, and for everything that you do. What I'd like to do is spend a few minutes giving the big picture of where we are in Alzheimer's disease today, and then talk about specifically what we're going to discuss with Dr. Brinton. I've been in this field for over 40 years. I remember the days when I started and nobody knew anything about it. We didn't know anything about the science of it, and people hadn't heard of it. And fast forward to today, it's obviously almost everywhere now. People are afraid of it. They're concerned about it. From a scientific point of view, I have to say that we've made amazing advances, and I don't use that word lightly. From a time when we knew nothing to a time when we have now a disease-modifying drug approved. Arguably, there's controversy around that. But what made that accelerated approval by the FDA possible was a brain scan. And the brain scan is able to detect the pathology, the amyloid, typical pathology of Alzheimer's disease, in the brain of living people. That brain scan was developed by ADDF as part of our mission when we started. We started funding the development of that brain scan in 2000. And that brain scan was approved by the FDA in 2012 as the first diagnostic test for Alzheimer's disease. It's also obviously the first biomarker for Alzheimer's disease, which is critical to drug development and why we invested in it. And it enabled the approval of this first drug, because what the drug does is, you probably all heard about the plaques and tangles. The drug removes the plaques from the brain, and we can see that on this brain scan, and that's amazing. The other advance that happened in just the last year or so is that we have an approval and on the market a blood test for Alzheimer's disease, which is also incredible, and it's going to revolutionize the way we care for people, because we can now go into a person can go into a doctor's office, a primary care doctor, and order this blood test that correlates exactly with Alzheimer's disease. So imagine you have, and can tell people with a high degree of certainty whether or not they have Alzheimer's disease. So imagine you have very early symptoms, and you're concerned, you can actually go in and get a blood test, and then maybe if you're positive, go into one of these clinical trials. And although there's controversy about this first drug that's been approved, there are 3 other drugs in the same class that are what we call anti-amyloid antibodies that have shown recently that they can remove that plaque from the brain, and that is correlating with some slowing of the disease. A modest slowing, but admittedly the first time that we've actually seen that, and it's such a major advance, it's really a revolution. We just came from the international meetings in Boston where we saw a lot of hope, a lot of data for the first time on the results of clinical trials. Our foundation, the Alzheimer's Drug Discovery Foundation, has 35 clinical trials around the world, and we fund internationally. We invest in biotech companies. We've made over 150 investments in biotech companies that have early stage programs in Alzheimer's disease, as we function as a venture philanthropy. And we've seen many returns, including the last couple of years, literally millions, tens of millions of dollars in returns on the companies and programs that we've invested in. The other exciting thing is really important, which is that we have a partnership with Bill Gates, Jeff Bezos, Mackenzie Scott, David Dolby, the Charles Schwab Foundation, and some others, Leonard Lauder. The partnership is called the Diagnostics Accelerator, and it's a $50 million partnership that's just been renewed by the funders for another $50 million over the next 3 years. What this is set out to do is now, although we have 2 biomarkers, and I mentioned the amyloid, we're also going to have a blood test for the tangles in the brain, and we just got approval for a brain scan that can detect the tangles in the brain, which are actually the dying neurons, and is going to be an even better test than the amyloid test in some ways. But the Diagnostics Accelerator is investing in accelerating these blood tests, in developing new blood tests that can detect inflammation in the brain and genetic changes of the genes in the brain that result in alterations in the way genes function, vascular disease in the brain, neurodegeneration of all different kinds. That's one class. New blood tests, digital technologies, like being able to do cognitive testing on the cell phone and capturing so much more data, so much more cheaply. This is the blood between the blood test and being able to capture cognition as an outcome measure in clinical trials is going to markedly reduce the costs of clinical trials. And when you consider that it's up to $2 billion to try to develop a drug, and $400 million for each phase 3 trial, and $200 million of that $400 million is just trying to find the right patients, we're going to have a big impact on the way clinical trials go forward, and we're entering into what I call the modern era of clinical trials for Alzheimer's. This is a major advance. This is where cancer was 30, 40 years ago. We're there now in Alzheimer's disease. The other thing, back of the retina, and just like this, because you can see those plaques, those amyloid plaques in the back of the retina, and just like diabetics go in for a retinal scan at their ophthalmologist or their optometrist, we're funding some companies out of Israel and other places that have a retinal test where you can actually see for about $100. The next time you go for your glasses or your glaucoma screening, you'll be able to go for screening to see if you have amyloid in the back of your brain, and that's also going to be an important way to accelerate clinical trials. Now, the last thing I want to say is that in this modern era, these biomarkers, combined with our understanding of the biology of aging, and aging is the leading risk factor for Alzheimer's disease, and there's many ways that we get to aging. So we have multiple targets. It's not just going to be the amyloid drugs. This is just the first of a new era of drugs. And so it's going to be combination therapy like we have for cancer. Cancer started out 50, 70 years ago. There was one drug, and then they added a second drug and a third, and now, God forbid, if you get cancer, you're going to be on 4 or 5 drugs, and there's going to be biomarkers to tell you what those cells that you have that are cancer cells are susceptible to so that your oncologist can design your chemotherapy for you. And that's what's going to happen with Alzheimer's. We're going to have those blood tests. Your doctor, whoever's taking care of you, your geriatrician or your neurologist, is going to design a therapeutic regimen for you that's going to be on multiple different pathways, not just amyloid, not just tangles, but inflammation, which is a hallmark of aging, and so many other drugs. Now, today, I'm delighted to be with my good friend that I've known for 40 years, Dr. Roberta Brinton, who is one of the leaders in the field in one of those targets that has been under investigation for many years, for many decades. And let me just tell you a quick story before I introduce Robbie. We met at the Rockefeller University. Robbie tells me we met in 1984. And the way we met was that Robbie was in the laboratory of Dr. Bruce McEwen, who was an expert and really one of the fathers of a science called the neurobiology of gonadal hormones. What does that mean? It means what is the effect of estrogen and testosterone on the brain? Imagine, and estrogen and testosterone have fundamental properties in organizing the brain during fetal development. So how many people have seen little girls and little boys? If you have, raise your hand, right? I mean, and how many of you have noticed that little girls and little boys are incredibly different from the day they're born? Why is that? It's because estrogens have a profound neurotrophic effect on the way the brain is organized during fetal development. And it differentiates, just like our bodies are male and female, our brains are male and female. But what that means is that as we go into adult life, we still need that. And then the question becomes if there are sex differences in the risk of Alzheimer's, and we know that women are at greater risk, but we don't know why, and we think it relates to estrogen. So I'd like to tell a story about how skills and different. And one way is that women are much better at verbal skills and social skills, and men maybe can throw a spear better and have some better visual spatial skills. And so I was told once that men have a daily vocabulary of about 35,000 words, and women have a daily vocabulary of about 70,000 words, and that's why when I come home from work, I've used up all my words, and I can't talk to my wife. So, we're going to hear from Dr. Brinton. Dr. Brinton is, she's going to tell you how we met, but she's a professor of pharmacology, neuroscience, and neurology, and director of the Center for Innovation of Brain Science at the University of Arizona in Tucson. Roberta.
Thank you, Howard. And thank you, Heidi and Tom, for organizing this. And thank you, Nancy. And I want to make mention that I am the Mel Goods Alzheimer's Drug Discovery awardee. Thank you. Yes. And this trip allowed me for the first time to meet Mel, and it was a great gift. So as Howard mentioned, we met at Rockefeller, and Howard was conducting a clinical trial in women with Alzheimer's disease and invited the Geek Squad, which were the basic scientists, to observe these women on this trial. And it was very, very informative for me, and I became particularly attached to one woman who has since passed and no longer living survivors. Her name is Dr. Rowena Onsbacher. And she was staying at the hospital, at the research hospital, and I would go in the evenings and would walk with her along the long esplanade of Rockefeller University, and she would regale me with stories of the feuds between Jung and Freud and Jung and Adler, and she was an Adlerian psychologist. And it was fascinating to listen to her stories in her professional life. One evening, I took her back to her room, and I bid her good night. I stepped out of the room and closed the door, and I waited 30 seconds. I knocked on the door and entered, and I asked, "Dr. Onsbacher, do you remember me?" And she was so lovely. She said, "I'm so sorry. Should I?" And what I learned from her is, first of all, she could not remember me for 30 seconds, but I've remembered her for over 30 years. What she taught me was that she never encoded me. I simply never existed, because she had code new information. What she didn't have is the ability to encode new information and to essentially establish that memory. I never existed. And that was a pivotal moment for me. Knowing one woman with Alzheimer's disease transformed the life and the career of one woman neuroscientist. And from that, because of Howard's research in estrogen and Bruce McEwen's, well, I have to say, I have to be truthful.
Don't.
When I was in Bruce's lab, it was like, I don't want to study estrogen. Estrogen, they speak in a language I don't understand. But it was Howard and Dr. Rowena Onsbacher that led me to change course in my career. And for the past 30 plus years, we have researched how estrogen works in the brain. And as I've mentioned to several of you, estrogen is the queen of Darwin. She has left nothing to chance. And part of that is the energy that the brain requires is quite demanding. And estrogen promotes that entire energy system. Now, just a little, background, and there'll be a test at the end. The brain weighs 2% of the body weight and consumes 20% of the glucose and the oxygen. From here down, we're a Prius. From here up, we're a Hummer. The brain is an energy hog. And that's why the decrement in the ability of the brain to generate energy can first be evidenced in doing what you are doing exactly right now. You don't know it, you don't feel it, but in your brain there's a whirling synaptic circuit that is taking what I'm saying and you're connecting it to what you already know. And then you're making new neural circuits, and you will remember me forever. Howard is over there. It's like, yeah, you will. But that's what happens, right? So you take in new information, you encode that information, you hook it up to existing information, and you make those associations, right? Now, that is the most energetically demanding function of the brain. And that is why it's the first function to be affected in Alzheimer's disease. There simply isn't enough energy to do that task. Later, those circuits don't exist, right? So that's what we see in brain atrophy, that we can see, with brain imaging. It promotes, back to the question of estrogen. What is estrogen doing in the brain? It promotes the entry of glucose into the brain and its conversion into energy ATP. As I said, it has left nothing to chance. At the perimenopause transition, there is a shift in estrogen availability. Now let me just stop there for a moment and give you a little bit of background on the perimenopause and the menopausal transition. So how many of you have gone through puberty? Right? That was a transition state, and it didn't happen overnight, right? It happened in stages. And for those of you with children, you've seen those stages, right? You've seen that this takes a couple of years, right, for this new capability to be integrated. And even though the brain doesn't look like the ovaries, doesn't look like the uterus, doesn't look like the testes, it is the organ of reproduction, right? The brain controls all of those organs. Now, what happens in perimenopause is, again, it's a transition state. And that transition state happens in stages. It's like a renovation project on an existing neural network. So just like any renovation project, everything looks good, except when you hear zzz, zzz, or you look up and there's water, on the ceiling. That, the little electrical problem that wasn't there before the renovation is there now, the plumbing problem. It's always a systems level problem. And that's what happens in the brain on these transition states. Most of the time, everything is fine. But in certain circumstances, essentially there's a component that doesn't come into play, right? Or there's an overreaction. And that overreaction is typically inflammation. One of the things that we have discovered over the past number of years is that it's really inflammation that kickstarts this whole process of this transition. And that kickstarts it in the brain, and then it moves forward. And so for some women and some men who undergo aging, that there can be this elevated inflammation in the brain. Okay. That elevated inflammation is a signal that the brain is not able to generate sufficient energy from peripherally derived fuels. Like, here comes the scary part. Bodies. Here comes the scary part. The scary part is that the brain can be its own fuel ATM, and it's called your white matter. That lining of the axons that contains lipids, and those lipids can be fuels. And that's something you really don't want to have happen, where you're utilizing lipids within the brain as a fuel source. Now, some of you may be wondering, hey, what about hormone therapy? Could this impact this whole process? And the answer is yes, it can. In most women, not all women. So hormone therapy, or estrogen therapy and progesterone therapy, can prevent the deleterious effects that can occur with a loss of estrogen. And to Howard's earlier point around aging, right? What estrogen really does is it delays aging. It doesn't prevent aging forever. It delays it. So essentially there is this sustaining brain function for a much longer time, and hopefully your entire life. So that's where I think it's important to think about when is appropriate to use hormone therapy and when it is not. It's appropriate to use hormone therapy both for women and men, different hormone therapy for women and men, when there are symptoms. If there are no symptoms, or that these symptoms have abated, then it's probably not a good idea to utilize hormone therapy. But for many women, they will experience hot flashes, which is the most common. They can experience insomnia, they can experience changes in emotional status, and they can experience depression. There are multiple symptoms of menopause, and they share one thing in common. They're all brain-related functions, right? And so that's part of why it's important to consider the impact of estrogen and hormone therapy on brain function, and to consider whether that's appropriate for you, and obviously in consultation with your physician. So I think we're kind of good at the moment. Now, that doesn't mean I'm done.
Mm-hmm. Well, Robbie, let me ask you a question. So there's this. So one difference between men and women is the menopause, and men have this gradual decline in testosterone with time and with aging and placement. Mm-hmm. But we don't have that. That's considered normal, and we give men testosterone replacement. Mm-hmm. But we don't have that dramatic change. And one of the reasons we started that estrogen trial in 1981 that you saw when you came and realized was that it was the time when we were learning about osteoporosis and using estrogen replacement therapy, because the same dramatic drop in estrogen at the time of the menopause leads to osteoporosis, and that dramatic drop has effects on the brain. So Bruce, our mentor, when we were sitting on rounds, and that was the whole purpose of the Rockefeller Hospital, to expose scientists like yourself to patients. And we were sitting on rounds, and Bruce said, well, why don't you give those women that were in the hospital estrogens? And that was like way from outer space. But when you put it in the context of where we were at the time with the treatment of osteoporosis, it made a lot of sense. And so we did the trial, and now it's 40 years later, and we're still trying to figure out the role of estrogen in Alzheimer's disease. So I want to bring it back a little bit to Alzheimer's disease. The dramatic, so we don't know exactly why women are at greater risk, but we think they are. It's like 1.5 times or something like that greater risk. And it's not just, as you've said, because the women live longer. It's something about the biology. And the obvious thing is menopause. And you and Lisa Mosconi at Cornell did some great studies, because if you think about it, the average age of menopause is 51, and the average age of the onset of Alzheimer's is 75. If you do the brain scans, maybe you can start picking up the amyloid at 65 or 55. but you did these studies with Lisa where you did brain scans on menopausal women and then looked at it in terms of how they were getting hormone replacement, if they were, if they weren't, if they were getting Prem-Pro or the natural 17-beta estradiol replacement therapy. And you found some really interesting things, which was a great experimental model in humans for us to try to link the menopause to the onset of Alzheimer's disease. And I think maybe could you tell us a little bit more about that work?
So thanks for bringing that up. So coming back to where Howard started, where women are at greater risk for developing Alzheimer's disease. They're at a twofold greater lifetime risk relative to men. And the thinking was, well, it's just because women live longer. And it's true that we do. We live about 4.5 years longer than men. And that doesn't account for what Howard was talking about, a 20-year, what's called a prodromal or preclinical phase to the disease, right? And what our work has shown is that it's not because women live longer than men that they have greater risk of developing Alzheimer's. It's because they start earlier. So if they're starting in midlife during this transition, right, then you wait 20 years, and sure enough, around 72, 75, women can be diagnosed with Alzheimer's, as can men. In the study that we did in humans, and what has really been so exciting about this work is that our colleague Lisa Mosconi and designed really want to work closely with us and designed the human imaging studies to match what we had done in our discovery science. And that was so important because then we were able to detect that beta amyloid can begin to form in the brain at the perimenopause and then increases with menopause.
And let me just say that the appearance of beta amyloid means that it's the appearance of Alzheimer's disease. It's the beginning of Alzheimer's. It's the classic hallmark of Alzheimer's. So what you're really saying is at the time of menopause and this dramatic estrogen loss, the cells are starving for estrogen, they're getting dysfunctional, and they're starting to degenerate, produce amyloid, and start the process of Alzheimer's disease.
Yes, and so one of the discoveries that we'd made a number of years ago is that estrogen actually activates a system that prevents beta amyloid being formed in the brain. So it actually prevents that. Okay. So what we found is that in women who had been on hormone therapy, so compared to perimenopausal women, compared to menopausal women, on women who had received hormone therapy versus those who did not, the women who received hormone therapy did not show beta amyloid accumulation in the brain, and they had greater cognitive function. We think that, from our earlier work, both in the discovery science and now clinical science, that the real drivers of the amyloid detection in brain is from women who carry the risk factor for Alzheimer's disease. They carry a gene called the ApoE4 gene. We all have the ApoE4 gene. We all have
the
two
ApoE gene.
Sorry, yes, the ApoE gene.
Yeah.
We all have the ApoE gene. Some of us have a variant called the ApoE2. Some of us have ApoE3, and some of us have ApoE4, and we can have combinations, one from each parent. Okay. So we think that it's really the women who carry the ApoE4 variant that are driving this detection of beta amyloid in this population. And why that is, it's critical, as Howard said, we're now able, based on ADDF funding, to develop these brain imaging capabilities. We're able to detect this very early, very, very early. And we're really excited because we have blood and plasma from these women, and we're going to, actually Lisa and I just talked about this last week, is test for whether blood-based biomarkers for Alzheimer's disease are showing up in these women.
So if I could just get us, so we could discuss whether or not women should or shouldn't take hormone replacement therapy with safety issues, but I'd like to take and what kind of regimens and so on, and the safety issues. But I'd like to get beyond that to your drug development program, which I know is your baby, and we've been funding it for many years now, actually, right? And it's a molecule called allopregnanolone, or as you like to call it, allo. And I know you've been at this trying to bring allo to market, and you started a biotech company, and you're finishing phase 1, as I recall. And
We finished phase 1 and we're in phase 2.
yeah, so be really interesting to tell us what allo is, and why you're trying to use allo for the treatment of people with Alzheimer's disease, not just at the time of the menopause, but actually people that are 75 and have Alzheimer's disease, and kind of a little bit about the process and so on, but really.
So I like to talk about this project because it started with ADDF and a conversation with Howard, and his advice to me was, cut to the chase, Robbie, just cut to the chase. Does it work? So that started a journey of several decades now. And allopregnanolone, or allo, A-L-L-O for short, is a molecule that all of us make. And we were exposed to high levels of allo during the 3rd trimester of pregnancy, when the brain undergoes a 4-fold increase in size.
The infant brain.
The infant brain. Yes, not the mother's brain. Your skull is formed. Yeah, no. You've got your skull already formed. So you don't want a 4-fold increase with a intact skull. So in the infant brain, undergoes a 4-fold increase in size during the 3rd trimester of pregnancy. And what's wonderful about allo is it's undergone one of the most challenging safety tests there is. It's safe in women of reproductive age with fetus. There's no greater safety challenge than that. And so we've been developing allo. We discovered a number of years ago that allo can promote the regeneration of neural stem cells in the brain, the stem cells that will make new nerve cells, new glial cells, new white matter, right? And so now we have finished up a phase 1 clinical trial that allowed us to test allo in 24 participants. It was a placebo-controlled trial, so a quarter of the people were on a placebo, and the others received 1 of 3 doses of allo. And the outcome of that trial was it's safe, in older individuals diagnosed with Alzheimer's disease. And we saw a signal of regeneration in the brain from MRI imaging. What we also effective in both, there was no sex difference. It was effective in both women and men. There was, however, a difference in persons who carried the risk factor, the ApoE4 gene, versus those who did not. It was effective in the persons carrying the ApoE4 gene. And it was not effective, no harm, no foul, but not effective if the risk factor wasn't there.
And Robbie, if I could just tie things together for everyone. One of the reasons you're interested in allo is it's in the synthetic pathway of these gonadal hormones, of estrogen, progesterone. It's a derivative of, maybe you want to talk about that a little bit, because that's kind of how it's related to the whole estrogen story, right?
Yes, yeah. So, yes. So there's essentially what I think is most important is that the human kind of chemistry, biology has been designed and tested for safety and efficacy. And these steroids, as Howard mentioned very early on, they have the impact to organize the brain system and to activate the system. So again, they're on both ends, developing, creating those neural structures and activating those neural structures. So we're very excited about the regenerative potential of aloe to regenerate the Alzheimer's brain, to restore the structure of the brain, to restore the function of the brain. We actually have several, we're recruiting a number of clinical trial sites here in Florida, with a number of them in Miami. We're hoping to recruit a clinical trial site closer to this area of Florida. But we're very excited that we should be starting to recruit participants in early 2022, January, February. So
Robbie, another interesting thing that you're doing is that you're developing a class of drugs called phytoserms, and you started a biotech company to develop them. Could you tell us why you're interested in phytoserms and what the company might be doing with them going forward?
So we started a company, New Therapeutics. It's NEU, like neural therapeutics, New Therapeutics, to both develop, once we have better data, more data on aloe, and now also a type of alternative to estrogen therapy, because many women have a concern of breast cancer, and so elect for breast health over brain health. And part of what we have spent, again, decades working on is creating an alternative to estrogen therapy that women can have both breast health and brain health. And specifically, this is they natural molecules found in plants, and they activate a type of estrogen receptor that activates estrogen action in the brain, but not in the breast. And so we're very excited about a clinical trial that we're going to be conducting to investigate. We already have data on safety. We also already have data on efficacy for hot flashes. And now we're really going to focus in on how phytoserms are impacting the energy system of the brain.
Yeah. I just want to open it up to your questions while we continue talking. If you have any questions, just please raise your hand and be happy to answer any of those that you might have. Yes?
You started to talk about the hormone, the type to take and the way that was most effective. Is there a certain hormone that you find to be the most effective and a certain way of taking it?
Can everybody hear the question? No. Yeah, so the question is about hormone therapy. What's the right kind of hormone therapy to take at the menopause?
So the answer is yes, we've looked at that. And we did a population level study to really start addressing, again, whether hormone therapy reduced the risk of developing Alzheimer's disease. And we found that it did, and that's replicating a number of studies that have already been done, published. Then we also looked at, well, which of these estrogen hormone therapies, because there are multiple products and multiple ways, as you mentioned, for delivery, are actually effective. And what we found is that the natural estrogen and the natural progesterone are the most effective. Again, Mother Nature has done her work.
17-beta estradiol would be the name of the chemical name of the product, and it comes in patches primarily.
Yeah, so you can receive them as patches or orally. And the data on the patches, because the patches are newer, there's less data. The benefit of patches is that when you take these hormones orally, like all foods, everything that's ingested orally goes to the liver first, and so and much of that molecule is broken down in the liver. That means you have to get a lot of estrogen or a lot of progesterone because a lot of it is broken down in the liver. That's the job of the liver. In contrast, when you take it transdermally through the skin, there's not this what's called first pass through the liver, and so you can use less concentration of the patches. That said, most of the patches are a combination of continuous estrogen and continuous progesterone. And I don't recommend that, the kind of continuous combined strategy, only because progesterone acts to mitigate some of the effects, the beneficial effects of estrogen. So I hope that kind of provides.
Yep, Bonnie?
Is there any connection about the age when women get their period and go into menopause related to Alzheimer's?
Related to Alzheimer's. So let me back up and step and say that interestingly, there are genetics around the onset of puberty and the onset of menopause that are comparable to your mother's kind of history. So there's some genetic component to that. Now, what I will say is that environmental pollutants can alter that. So there's that. And then with respect to Alzheimer's disease, interestingly, the individuals who have received oral contraceptives, earlier in life, basically the longer one is on estrogen, the greater the impact on risk reduction, right? So later menopause is associated with greater reduction. And pregnancies are associated up until five pregnancies, and then it's kind of not as beneficial.
But women who have a delayed menarche also tend to have a delayed onset of menopause. People who live to be 100, those women, everything is delayed, so they have delayed, instead of having menarche at 13 or 14, it might be at 17 or 18 that the periods start, but then their menopause is at age 60, they live to be 100 and maybe don't start getting cognitive impairment till into their 90s. So there's something about the estrogen exposure and the timing of that. There's also just been a recent study, and there have been several along the way, that things like early menopause is a risk factor for the development of Alzheimer's, like surgical menopause.
Yeah,
women
yes.
who have their ovaries out 10, 20 years before, before age 51 have a greater risk of Alzheimer's. So it does kind of point to this idea that the total lifetime exposure of endogenous, of natural estrogen is correlated with the time of onset of Alzheimer's disease. Was there another question? I forgot. Yes.
So I do want to make sure that the men realize that we haven't forgotten you. We're actually studying the male aspects of risk for Alzheimer's. And the female was a smart idea. And studying the female, now let me just tell you why studying the female was a smart idea. It was not a feminist idea, although it could have been. But it was smart around the biology because we, in women, have this age-locked transition, midlife aging transition. And as Howard said, that doesn't happen in the male. That essentially it's this slower decline in androgens over the lifetime, or not. I mean, some men can maintain normal levels of androgens throughout their lifetime. What we've begun to ask is, what happens? We know that cancer increases with risk of cancer increases with age, and we've begun to ask, what happens if women are put on anti-estrogen therapies, these selective estrogen receptor modulators, or men are put, on prostate cancer therapies and anti-androgens for prostate cancer. And what we found for women is that tamoxifen, as well as aromatase inhibitors, are associated with about a 25% reduction in risk. That's relative to hormone therapy, which is about a 50% reduction in risk. It's not absolute, right? but it's about 50% reduction in risk for developing Alzheimer's disease. Whereas breast cancer therapies, specifically tamoxifen and the aromatase inhibitors, about 25%. So they're acting like an estrogen receptor antagonist in the breast and the uterus, but acting like an estrogen, a little, like a modest estrogen in the brain. What about men? So first of all, testosterone works a bit like estrogen does in the female, in the sense that higher lifelong testosterone is associated with reduction in risk of developing Alzheimer's disease. So that's part one. The bad news is that prostate cancer therapies, anti-androgens, are associated with an increased risk of Alzheimer's disease. And that's in part because of a therapy that's common to all of the combination therapies. And I can talk more about that later. But we do have one, and this is not yet published, so I'll wait till it's published by peer review to talk more about this. But we have one molecule that looks promising that can mitigate some of the adverse effects of, I've forgotten you guys, agonists. But just to let you know that I have not forgotten you guys, we're on to you now.
Yes, sir.
Dr. Brinton, you had mentioned that you have some clinical trials that are going on, and possibly in Miami, and you also indicated that there could be some that were in this area. Can you be a little more specific on that? And if somebody was interested in clinical trials, what should they do? Who should they contact? And are you looking for people that are already showing symptoms, or maybe just women that are experiencing menopause?
So thank you for asking that. So the sites that we have in Miami and the one site that we're looking at closer, to Palm Beach, are not yet recruiting participants. You can go on to the Center for Innovation. We will have a website, probably in about 2 weeks. The Regen Brain, should be easy to remember, Regen Brain, all one word. That will then begin to give updates on sites that are recruiting. We will be recruiting people who are diagnosed with mild Alzheimer's. And that's both men and women. So that's going to be our New Year gift to everyone.
Any other questions? Yes.
Yes.
We'll get to you.
I'm going to let Dr. Phillips talk about that.
Yeah, so the question is about the new drug that was recently approved for Alzheimer's called Aduhelm. It was developed by Biogen. What was controversial about it was that mostly around the process of how it was approved and the ambiguity in the data that was submitted for that approval. In terms of the ambiguity of the data, there were 2 clinical phase 3 trials that were submitted as part of the FDA application called 301, 302. And 301 was actually was initially reviewed as a negative study. In other words, there was no effect seen. And back in March, the trial was actually stopped because it looked like there was no effect. However, during the time between the time that the committee reviewing the trial and said that it should be stopped and the time it was actually stopped, more people were enrolled in the study and more data was gained. And then about 3 or 4 months later, the new data was reanalyzed, and it looked like there was a signal, particularly in the people that got the highest dose. The 302 trial was quite positive. And so you had kind of really one negative study and one positive study, one study that was kind of hard to interpret and one that wasn't. The problem with the process was that generally the FDA is very independent in its review of pharmaceutical drugs, and the big to summarize the data and present with the FDA people to review and present, to summarize the data and present it to an external FDA committee. But in this case, for whatever reason, the FDA director in charge of the neurology division actually met with the Biogen people internally to the FDA, and they sort of prepared this presentation to the external expert committee together. And then when the program was presented to the external committee, the neurology director was really sort of hyperbolic about his opinion at the introduction of the committee meeting about the value of the drug. And that was seen as kind of really biasing the whole day. He used regulatory words that would be used to say this drug is approvable. When the statistician who analyzed the data for the FDA, however, said that, and this was a very sophisticated, many months of analyzing the data, said that there was no evidence for efficacy in the data, while the company argued that there was. So there was a disconnect between the neurology director, who had been really working with the company, and the statistician for the FDA, who had a free voice to state their opinion to the committee, to the external committee, and said there was no evidence for efficacy. The committee, 11 people, reviewed the data from the day and the whole day. I won't describe it, but 10 of the 11 members on the committee said that the drug should not be approved. It should be rejected. And one person abstained. Subsequent to that, 3 of the committee members resigned from the committee because they thought that the FDA mishandled the whole process, and also because the FDA ultimately gave the drug what's called an accelerated approval. Now, this is a mechanism that the FDA has to approve primarily drugs for cancer, where you have a tremendous unmet need, people at risk for dying, and you don't have clear evidence of efficacy to get a full approval, and you don't want to disapprove the drug because there's some signal there, so you give it an accelerated approval, which is kind of like a tentative approval. It's demand of the company that they'll approve the drug, they'll let it go to market, but the company has to do another trial over the course of the next 9 years, okay? So meantime, people are getting the drug and companies making a lot of money and so on. But that accelerated approval is based on a biomarker. In other words, in cancer, it might be the X-ray of the lung, of the tumor in the lung, or some biological biomarkers. In the case of Aduhelm, as I mentioned earlier, this brain scan that we developed was used not only to enroll people in the trial, but to show the efficacy of the drug. And the drug had great pharmacology. It removed the plaques in over 80% of the people at about 12 months. So it clearly worked. But the leap that the FDA made in giving it an accelerated approval was to say that because the drug removed the amyloid from the brain, it was reasonable to expect that that was linked to cognitive improvement, to slowing of the disease. And that evidence to proven in that trial. It was suggested in that trial, but there wasn't really evidence to prove that it's like you take a statin and you lower cholesterol, and you can be pretty sure that if you lower cholesterol with a statin, it's going to prevent a second heart attack. That's widely accepted. But it's not widely accepted that if you remove amyloid from the brain, that you're going to slow the rate of decline of Alzheimer's disease. So that linkage wasn't there, but the FDA, after they went back and kind of closed behind closed doors, they came out with this accelerated approval, which is not a rejection, it's not an approval, it's like you have to go and show us over the next 9 years that the drug really works. And of course, so what happened is that it unlocked the door of the drug to get to market. Now, the problem is the other controversy around it is that there's real safety problems with this drug. 40% of people that have this ApoE4 gene that we've talked about today who are at greater risk for Alzheimer's disease had brain swelling. And we know that that's kind of manageable, but it's a serious side effect. And 20-30% of people that didn't have ApoE4 also had brain swelling, and they also get some hemorrhages. So there's a lot of burden to taking this drug, which is another clinical controversy because it's an IV infusion every month, takes about 2 hours, and the last controversy about the drug is that Biogen priced the drug at $56,000 per annual dose for the course of the drug for a year, 12 doses. And then to add on to that, you have to add on the cost of getting the drug at an infusion center. So all the nursing, the medical technology, all the infrastructure to go to an infusion center to get the drug. It's estimated that the drug would cost $100,000 per person. Now, we just read that Medicare just raised its budget for its Part B pharmacy costs, which is where this drug would come out of the Part B of Medicare, by $21 billion. And the reason is that Medicare is anticipating that that's going to be what the government is going to have to pay to people who ultimately get this drug, even though it's not clearly effective. So it's already had an impact on Medicare, which goes back to essentially us as taxpayers to be able to pay for this drug. So we really, this is what's going on now. It's kind of driven by unclear science. I have to say that although the Biogen drug wasn't entirely clear, we were just at the international meetings and there's been a lot of press releases by Eli Lilly about the drug that they have that works in a similar fashion to remove the amyloid from the brain and seems to be much better. And the thing about that is they also use the brain scan that we actually, full disclosure, we get royalties from Pennsylvania on that brain scan because we're a venture philanthropy. But the Lilly drug is slightly different chemically. It seems to be more effective. It decreased the rate of decline in patients by a greater amount. There's a delay that we could predict to institutionalization and quality of life and all these other real-world improvements. And I think the importance of the Lilly data, that it's starting to look, a Japanese company called Eisai now, and a fourth company, Roche Genentech, that it's starting to look like this approach does work. But it's not a huge effect. There are side effects, but it looks like it's the first disease-modifying drug for Alzheimer's disease, and that's huge. And that's how cancer started and diabetes started. You know, we started with insulin, and now diabetics are on 3 or 4 drugs. Hypertension, people are on 3 or 4 drugs. Cancer, 4 or 5. This is the first, and I think it'll probably go forward despite the controversy. I think the really sad part probably is, at the end of the day, the pricing of the drug. And we're hoping that when Lilly's already applied to have their drug approved by the same mechanism, accelerated approval, where they'll be forced to do another what's called phase 4 study. But I think we're hoping that they won't make the same mistake on the pricing and they'll make it more available to people, because if the drug costs $100,000 a year and it's a Part B drug, unless you have Medigap insurance, it'll cost you $20,000 a year to get the drug. And not many people in this country have $20,000 to give a drug that, you know, doesn't have a huge effect. It has an effect, but not a huge effect.
So the detail about what you were just talking about, you said that drug gets rid of amyloids and treats amyloids, but there's 2 components to
Tangles.
plaques or the tangles and the amyloid.
Yeah.
So if it gets rid of one and doesn't get rid of the tangles.
Actually, it's a great question. So what we think, in this theory of the amyloid, is that the amyloid is representing some sort of neurodegeneration, and this molecule, which is real sticky and neurotoxic, gets out, right? And so the theory was that this neurotoxic molecule that accumulates in the brain in plaques is neurotoxic and leads to the tangles. Okay, so what we just saw on Friday for the first time was data from the Biogen study, that because we have a blood test for the tangles essentially, and we have a spinal fluid test for the tangles, and we also have a brain scan for the tangles. And what Biogen showed on Friday is interesting, that the Aduhelm not only removed the plaques from the brain, but it reduced the biomarker in the blood for the tangles. So it looks like it's actually, there's a biomarker for showing that it's reducing the rate of neurodegeneration, as represented by the tangles, by this blood test that measures a molecule called phospho-tau. Does that make sense? Yeah.
So I think the short answer is that the short answer is removal of beta amyloid appears to also impact the removal of or the generation of beta or the neurofibrillary tangles, right? So suggesting that there is a relationship between beta amyloid generation and neurofibrillary tangles in the brain, right? So it's, again, as Howard said, it's a leap forward. Both of these effects are missing the disease. I think that they're not going to be game changers in reversing the disease. I think they're going to be game changers in slowing the course of the disease. And you can imagine now, let's say we add a regenerative therapeutic, right? So you're giving the brain a fighting chance, I think would be one way to positively look at this.
But Robbie, that would be with allo, but just to be clear, I mean, the study that we did way back 40 years ago of estrogen was for women that already had Alzheimer's disease, but we're not recommending that women that already have symptoms or even more than, let's say, 5 years out from menopause, right? The horse is out of the barn by that time because the loss of the neurotrophic effects of estrogen are sudden, they're dramatic, they set off a process, but then somehow women adapt to it and most women get back to normal, right? But we're not
They
recommending
get a new normal.
a new normal. But
It's
we're
a
not
new normal.
Right, and women have successful lives, obviously,
Yeah.
postmenopausal, but we're not recommending that women, let's say 55, 58, that are 5 or 7 or even 8 years out of, or women 65 or 75 that have Alzheimer's diagnosed, that we would treat them with estrogen. You might want to, as you say, be treating them with allo, which is, I guess you would consider it's a neurosteroid, but it's not necessarily a gonadal hormone, I suppose.
No, and we find that that the Alzheimer's brain and the aging brain can still regenerate.
Right.
So we're very excited about that.
And there are other things that regenerate. For example, exercise has been shown to promote neurogenesis, which is quite interesting. And that is a major finding recently. Okay, we have a question in the back.
I think you partially answered the question I had, but let's say that a woman is 75 years old and doesn't show any signs of Alzheimer's at this time. Would you recommend hormone therapy? And if not, why not? And the same question with respect to men.
Yeah, I mean, I think that, as we just said, that prescribing hormone therapy outside of the menopause, we have to weigh the side effects of potentially uterine cancer. It can be monitored. Women can get mammograms, and we can do ultrasound on the endometrium to look for endometrial thickening from estrogen replacement. But for example, we used to think that estrogens prevented heart disease. That data came from a lot of epidemiological studies, but now we're not so sure, and the large clinical trials prove that actually, in some ways, estrogen replacement therapy caused more strokes and wasn't valuable for preventing heart disease. And I think, again, it's this critical period. It's the same thing as osteoporosis. If you treat osteoporosis at the time of the menopause when there's a dramatic increase in bone loss because of the lack of estrogen effects, and you treat it preemptively, then you can prevent osteoporosis. But if you wait 3 or 4 or 5 years post-menopause and give estrogen, no benefit. And so a woman that's post-menopausal, already lost the bone, there's no benefit. And so I wouldn't recommend it. And it's not just the risk of endometrial thickening and cancer or breast, but also the increased risk of stroke and other forms of clotting in the elderly woman. I think that would be too much of a risk factor. Bonnie, I think you had a question.
I'm very curious. So if this new drug, which helps to lessen the tangles and the plaque, that won't reverse a person already diagnosed with Alzheimer's. So what can you do? What do you hope to be able to do to reverse the disease?
Right.
Other than getting rid of those two elements?
So the question is, what can you do to reverse the disease? Well, let me just say 2 things. One is we're slowing it down. So let's say a person is 75, and I just was involved in a study actually about this. Let's say what you want to model is what's the benefit. So if you had cancer and you watch TV and you see these amazing ads and you read the very fine print at the bottom, which you can't really read, it actually says, although everybody's smiling, it actually says, you're going to gain 12 months of life from this. You're not going to live forever. You're going to gain 12 months. But people value that, right? They value that extra 12 months of life, even though their hair might be falling out or whatever. Looking at this drug and modeling what's the benefit, we see that with Biogen's drug, which is not the best one in development now, that there's a delay to progression to the next step of about 2 or 3 years, let's say, to moderate dementia, where people are really functionally impaired and have to get care. So let's say you're 80 years old and you have mild cognitive impairment, you're still functional, and I give you this drug, and instead of saying it's going to change the rate of decline by 35%, I say to you that you're going to be able to play with your grandchildren and recognize them and, go shopping or whatever for an extra 2 years. That could be pretty meaningful to a 78-year-old who has a life expectancy of 6 years normally, and what I call a functional or a cognitive life expectancy of 3 or 4 years. Now I'm extending that cognitive or functional life expectancy by 20-30%. And so that's meaningful. That has real world value. Does that make sense? All right.
Good evening. Thank you so much for this fascinating and insightful presentation. My question is if you could speak a little bit more about the accelerator and its strategy for deployment, because ultimately we have a really fractured and fragmented healthcare system.
Sure.
And so across the country, hundreds of thousands of doctors, primary care physicians, ER physicians, optometrists, ophthalmologists. I have yet to hear in my own circle of care team any of these diagnostics as being either offered or when I've personally asked, what can I do now? Because I have history in my family and because I like to get a baseline, nobody has proffered anything to me that I could say is
That's a really exciting question.
discussed in this accelerator program.
Yeah, that's a really exciting question. And the reason I find it exciting is there's so much going on that's going to come to market in the next few years. It's just a revolution. So we just had the first blood test approved as a CLIA-approved test, which means that the company called C2N, which we've been investing in for about 7-8 years, I've gotten results. I've had my patients get the blood drawn. It goes to a specialty lab in Washington, St. Louis, and I got the results back. One patient had Alzheimer's, the other didn't. This was just approved a few months ago. The company is just rolling out the test. They're just starting to market the test. And then that's an amyloid test. The next test that'll come to market, which will be even better in 2 or 3 years, maybe a year if we're lucky, is a tau test that reflects the tangles. The retinal test will probably come to market with a CE mark approval in Europe in the next year or so, and then a 510K here in the United States in about a year or two. So we're just on the cusp. If you're in practice, you're going to start to hear about and see these tests in the next, I predict, in the next 1, 2, 3 years. And you can, if you want to use the blood test for Alzheimer's disease that's on the market, the name of the company is C2N. I can put you in touch with the representatives that's working with doctors all over the country, except in New York State, because we have the most strict rules for approving tests, and it's not available. The only state it's not available is in New York. But I could put you in touch with the representative of the company, and she'll work with you on, you got to put it on ice and draw the blood in a special tube and so on. But you can start ordering it, and they're rolling it out now, so they're just starting to market it. And it's going to change the world. I really believe that, not only the clinical trial world. So it's just an incredibly exciting time. These digital technologies, we're seeing that with the Apple Watch, right? And you heard about Apple's going to work with, I forget who now, Biogen. Yeah, Apple's working with Biogen to roll out tests on the Apple Watch. And you can already use the Apple Watch for, doing your digital health, like if you have if you have an arrhythmia, your watch will tell you. So we're just entering this era of using technology to monitor health, and we're going to see it not just in heart disease, but we're going to see it in Alzheimer's disease and brain health in a big way. And it's so you're right, you haven't seen it yet, but I predict you will see it in the next, probably in the next year or 2 or maybe 3, something like that. You're going to see it, there's no doubt.
So to expand on that a little bit, so part of what, Howard is referring to is, for example, our work, both in the estrogen field and regenerative therapeutic for Alzheimer's disease, has been decades in the making. And we're now about to launch to the public the outcomes of decades of research, right? That, yes, there's been a lot of focus on amyloid, and there's been commercialization in, amyloid therapies. And at the same time, there's been, thousands of scientists and clinicians working on therapies that are not amyloid related, right? That are going to really, I think, advance precision medicine for Alzheimer's disease, that we're going to be able to identify the risks that a person has and to connect that to their gender. We're going to be able to connect that to their stage, whether they're pre-Alzheimer's, whether they're in the early stages, whether in the mid stages, and really create a personalized and precision approach to Alzheimer's. And we are literally, I think, 5 years away from beginning to be able to do that. But it's within our lifetime, which is really very exciting. It's within our lifetime.
And just getting back to your commercialization, part of the actual mission of the accelerator as a nonprofit is to accelerate the commercialization of these drugs. So we're not kind of funding basic research around the world on new tests. Sort of exploratory studies are not within the realm of what we're doing. We're working with Roche to accelerate the development of their test. And it's just about there now, and they have 70,000 placements of their technology around the world. So as soon as that assay is ready for approval, it's going to be deployed all over the place. And we're working with other companies to do similar things now. So we're very focused on the commercialization part of this. That's where we think we can have an impact to accelerate this to market. Yes?
It's wonderful to hear all these fabulous therapeutics that are coming up and the science that you're involved with. But you haven't spoken about prevention at all.
Yeah. Well, I'll give you a couple of examples. And prevention comes in different forms. So one of the main forms that we're realistically working with in the model of how we as a foundation can accelerate prevention is this whole idea that the first symptoms of Alzheimer's are a stage called mild cognitive impairment, where people just have memory problems, but they're basically functional. They don't, they might have some problems, a little bit of problem with scheduling or managing their finances, but they don't need help with, eating or any of that kind of thing. It's a very early stage. And so one form of prevention is preventing the progression of people with MCI who would have positive amyloid scans, for example, and well biomarker evidence of Alzheimer's disease from progressing to the first phase of dementia, which is called mild dementia. So 80% of the clinical trials that we're funding are prevention trials, what we call secondary prevention, to prevent people with MCI to get to dementia. Now, to say biomarker evidence, like they have, and find normal people who have, let's say, biomarker evidence, like they have positive brain scans and no symptoms, and prevent them from getting symptoms. Those trials are underway. We're not necessarily involved in all those because those trials, the trials that we can afford, that we can support from MCI to mild dementia, we could do that for maybe $5 million, something like that, or part of a consortium. But the trials where you want to prevent somebody from going where they just have evidence of the disease in the brain and going, to try to prevent 5 or 10 years later the onset of symptoms, those trials cost hundreds of millions of dollars. And there are some of those being funded by a consortium of biotech companies, pharmaceutical companies, and government. But one of the things that I'm really excited about is we're funding a trial called FINGER, and that stands for the Finnish Geriatric Study of Prevention. It was originally started by Dr. Mia Kivipelto, a geriatrician working out of Helsinki, who's on our board now. And Mia actually, so we know from prevention studies that 40% of the cases of Alzheimer's disease could be prevented by things like exercise, diet, don't smoke, don't drink too much alcohol, sleep all right, manage your diabetes and hypertension, those kinds of factors, hearing loss. There's about 12 of them that they've identified. And the very prestigious commission in the UK came out with a paper not too long ago that if somebody followed all of these, they could prevent 40% of the cases. What Mia did is she took these items and she packaged them into a randomized clinical trial. So one group kind of got the education that if you did these things, you could prevent Alzheimer's, but they got no follow-up in clinical practice for compliance, so that month by month by month they actually did it. Whereas in her treatment group, so to speak, her intervention group, people were followed very carefully and encouraged to follow the advice of the doctors and their lifestyle and their management. And she showed that with the compliance intervention, if people really followed the rules, she could slow down the progression of cognitive impairment by several years. Now, if you think about heart disease, it's very similar to preventing heart disease, the interventions, exercise and so on. But then we add statins for primary prevention of your first heart attack, right? So you combine the lifestyle with the comorbidity management, diabetes and hypertension, with a drug that's specific for that disease. So what we're doing, we gave Mia recently a $3 million investment to add metformin, which is one of the leading diabetes drugs, to her prevention trial to see, because metformin is really the leading anti-aging drug. We funded a clinical trial of metformin for Alzheimer's disease, showed that it had effects on biomarkers and metabolism in the brain, and there's a very big study going on now by Novo Nordisk of a leading diabetes drug, and we could talk about diabetes and Alzheimer's for a long time, but the point is that metformin was the first choice to add a drug to this lifestyle and comorbidity management to see if we could have even a bigger impact on preventing Alzheimer's. So there's a lot. One of the missions of the ADDF, talk longer, I think we kind of come to the end of time, but prevention is one of the missions of the ADDF for sure. I think the kind of studies that we talked about earlier, getting back to the context of estrogen, is how do we design doable studies? And Robbie's study of looking at brain scans in menopausal women and finding out that women who are going through a natural menopause are starting to accumulate amyloid, but if they take estradiol, they don't accumulate amyloid. That's an incredible finding. I mean, we can't prove that 20 years later people aren't going to come out demented, but to know that they're not laying down amyloid, which is the beginning of Alzheimer's disease, I think is a really, it was really an innovative approach to doing prevention research. I think we have to stop now, if I'm not mistaken, Deb. Thank you very much for your attention. We really appreciate it.
Thank you.
Thank you, Howard. Thank you, Robbie. And thank you all for being here this evening. And thank you again, Heidi and Tom, for making this series possible. We're incredibly excited to be coming back down here in January. On the 31st, we'll have Dr. Frank Longo. And then on April 26th, we will have, let me tell you, Dr. Jeffrey Cummings. So as you're leaving tonight, you may notice that the Royal Palm Bridge is now lit up purple, which we did in honor of Alzheimer's Awareness Month and also to announce this new series. On your way out, if you want to pick up there are brochures about our next two speakers for the series, and also please remember to save the date for our Hope on the Horizon dinner taking place March 3rd, honoring Jane and Aaron Lauder. Thank you again for being here, and we wish you a very happy holiday season.